This is Dr. Heidi Wolburn, and you are participating in Biotechnology and Society, Bio 104. As we begin this course, I want you to think about what exactly is biotechnology. I find one of the most useful ways to understand what biotechnology is to break the word apart into its root words. So biotechnology is derived from the following words: bios, techos, and logos. Bios, which is where we also get the word biology from, is the study of life. Techos, you can think of the word Pentateuch, for those of you who are biblical study majors, just means the 5 books or the 5 tools. So techos is actually means tool. Logos, you might remember from the book of John, also means the word or the study of. So it's the study or essence of. So you put all this together, you can understand that biotechnology is the study of tools from life, or the study of living things. And really, if you want to think about biology, it's concerned with questions about natural phenomenon. And technology is able to take the answers to those questions that we might have and attempts to solve human problems and various human needs. From a historical perspective, we would like to know, when did ancient biotechnology begin? Thousands of years ago, there were nomadic people that survived by living in various mobile camps, and they would hunt wild animals, collect wild plants, seeds, and nuts for immediate consumption. But it was really the establishment of agrarian societies, which was about 10,000 years ago, when people began domesticating plants and animals. And this was taking place simultaneously in various parts of the world, predominantly in the Near East. We know that in Iran and Iraq, Turkey, Syria, Jordan, Israel, they were all cultivating wheat and barley. We can look at ancient collections and see that seeds were being collected from wild plants for cultivation, as well as the domestication of wild animals. We also, in various writings, have been able to determine that various forms of artificial selection were taking place. Farmers were collecting plants with the most desirable traits and would set them aside for planting the following year. Farmers would also breed the most prized animals, and we even have examples of this in Scripture, as demonstrated by Jacob in Genesis chapter 30, verses 31 and 32. We know this to be true as well just by the evolution of corn, dating back from 5000 BC to AD 1500. You can see that progressively the corn was getting larger and the kernels were getting larger, just based on farmers collecting the seeds, specifically from the corn that grew the largest, most robust, and healthiest husks. Here we have an ancient artifact dating back nearly 4,000 years ago, which is made from plastered and painted wood, demonstrating a bakery of sorts. You can see the women on the left have taken various grains, which were subsequently ground into a flour for dough. The gentleman in the middle would then add some sort of water and make it into a loaf, and the gentleman on the far right would then bake the bread, and this would be similar to a pita type of bread. We know around 1800 BC, Egyptians started to realize that old, uncooked but fermented dough could be used to make a new batch of dough. It's thought that this process of fermentation was discovered quite accidentally, but leading to the more common types of bread that we have today. What the ancient Egyptians and also the ancient Chinese cultures were doing was to begin to harness this process of fermentation. So, as in the case with the fermenting bread in the slide before, what was happening was yeast cells, shown here as Saccharomyces cerevisiae, were taking the sugar or the starch from the grain, and they were converting that into CO2 and ethanol. So rather than developing a flat bread, which is the pita bread, they now had fluffy bread, which is demonstrated by the trapped CO2. So when you open up a crusty loaf of bread and there's lots of air pockets, that's because lots of CO2 was produced by the yeast in the bread. The ethanol that's produced during this process is simply baked off during the cooking process. So the ancient Chinese, they were incredible at using different types of microorganisms to produce different byproducts for their various needs. So they would use specifically certain types of microorganisms to produce lactic acid, which would be necessary for the fermentation process in the development of soy sauce, for instance. And they would use other types of microorganisms, such as Escherichia coli, to produce acetic acid, which was necessary for the production of various types of vinegars. So as we define ancient biotechnology, it needs to include this idea of fermentation, which is simply a microbial process in which enzymatically controlled transformation of organic compounds occur. And these different microorganisms that can foster fermentation include different types of bacteria as well as yeast. And we can see that even today in all the various types of food products that we have that include both this process of fermentation as well as the preservation of various types of foods using brines and salts. So as I mentioned earlier, we have examples of the Chinese cultures using various types of bacteria to produce specific products such as cheese and yogurt versus wine vinegar. So a full definition of ancient biotechnology should include the following: the planting of crops, the domestication of animals, and the fermentation of organic matter. Now we'll move from our definition of ancient biotechnology to a classical definition of biotechnology. This is really based on the work of Louis Pasteur as well as Anton von Leeuwenhoek. Leeuwenhoek, in the late 1600s, built his own microscope, and he was curious in nature and proceeded to look at everything that he possibly could under his self-made microscope. One of the things that he looked at was fermenting beer. And he drew the following picture, which he did not know what that was, but this is what he saw. We now know that what he saw was yeast cells in the fermenting beer. His studies were largely forgotten until about 200 years later, when Louis Pasteur decided to try to understand what happened during the process when wine would ferment and spoil. Pasteur understood that as wine fermented and as it was aged, it would oftentimes spoil. So what he ended up doing was to develop a process by which he would allow the wine to ferment and produce the alcohol, but then he would go ahead and heat that mixture, which would subsequently kill the yeast cells so that it wouldn't actually spoil, which was due to the production of lactic acid. So this led to a whole new industry, which was utilizing industrial fermentation. So in terms of classical biotechnology, we can define it as utilizing living organisms to make a product or to run a process, such as was the case with industrial fermentation. A more modern definition of biotechnology, which would be used today, is really the use of any tools or techniques which use living organisms or substances to make or modify a project to improve the lives of plants or animals, or to develop microorganisms for various uses. So when we think of biotechnology, this can include things like recombinant DNA technology, gene transfer, moving genes from different types of microbes, from microbes into plants and animals, the manipulation and transfer of animal embryos, as well as stem cells, genetically modified organisms and plants, as well as the production and scale-up of various types of recombinant proteins and monoclonal antibodies. The biotech industry, as we know it today, really emerged in the early 1970s with the discovery of recombinant DNA technology, which we will discuss more in this course. Today, it's responsible for over 200 therapies that are presently on the market. There's over 1,400 biotech companies, and really it was the production of recombinant human insulin in 1982, which was the first approved biotech product made in collaborations between Genentech and Eli Lilly, which launched this entire industry. In summary, you should come away from this short PowerPoint presentation and understand the evolving definitions of biotechnology from a historical perspective. And you should also be able to come up with a list of several examples of biotechnology. In conjunction with this PowerPoint presentation, there's a series of readings that have been posted on Canvas in the module entitled The History of Biotechnology. Once you've completed those readings, please answer the homework questions. That can be downloaded as a Word document. You can then type in your responses and resubmit that to me through Canvas. I look forward to working with you over the course of the next several weeks in Bio 104, and I hope that I can get to know you better and learn from you through the process.